starting on slide 11, chapter 2. The more of one particular substrate that's available to the body or the cells around it at a particular time, the more those cells are going to use that particular substrate. The more you have something, the more you want it. It's called the mass action effect. The other thing that is a limiting factor in terms of the regulation of carbohydrate, protein, and fat is the amount of enzymes that you have. Enzymes break down molecules and help catabolize molecules so that ATP can form. One of those molecules is ATPase. It helps break down ATP into ADP and allow for the energy release. You look on figure 2.2, I believe that is slide number 13. If you look at the left, it seems as though the energy state of the molecule, or the activation energy of the molecule, has a bigger hump than on the right. You can think of activation energy as almost like a boiling point. If the boiling point is higher, you have to pump more energy in to get the water to boil. But if you can lower the boiling point, then it doesn't require as much energy to keep the water boiling. So an enzyme or something like creatine kinase there is important because what it does is it lowers the activation energy, so it makes it so that the chemical reaction is more prompt, or takes a little bit less to get going. Each of the pathways of the breakdown of the fuel, as the fuel gets smaller and smaller, requires another specific enzyme. The most important enzyme in the whole breakdown of a fuel or process is called, that process is rate limiting enzyme. And in the case of the breakdown of carbohydrate is in the macro nutrients, it's one of them is creatine kinase. Figure 2.3 on slide 15 just shows that one of the things that happens when you start to break down the fuel is that there's a release of energy, free energy. There's you have the fuel, you have the ATP comes in, it's in its activated state as a fuel, and then there's an enzyme number 1 in that arrow, and then the enzyme makes the ATP break down into ADP plus that phosphorus ball, and then the phosphorus is free to roam in the body for gluconeogenesis, or it actually just going to combine back to the ADP, eventually make ATP. And in the breakdown of ATP, there's free energy released, and there's also heat. That's why, like, when you run, your body temperature goes up, because not only does it burn, does it break down ATP, so you can use that free energy for the movements of the actin, myosin cross bridges, but there's also a release of heat. And that's why shivering works. Like when the ATP breaks down very quickly, there's a release of heat and it makes your body warm. So also there's a byproduct. There's like a break, as the ATP breaks down, there are byproducts. And those byproducts, they start to build up as each ATP molecule breaks down. There's more and more byproducts and more byproducts. And then when the amount of byproducts, probably ADP and extra phosphorus, start to build up and build up and build up and build up and build up, then that makes a signal for the rate limiting enzyme to release and break down ATP more. So that's called like a negative feedback loop. Once one thing starts to pile up, it acts as a signal for the rate limiting enzyme to start the whole process over again. Now we're on, there's an animation on slide 16, there's a video on slide 17. So slide 18, we're at high energy phosphates. ATP is stored, but it's not stored in massive quantities in the body. It's stored for a very small period of time, and then ATP needs to combine with water and ATPase, which is that energy, which is that enzyme, and that when they do, it breaks down to ADP plus phosphorus and energy. That process, because water is needed, that process is called hydrolysis, where the water is lysed, is broken apart. So ATP will break apart water and combine with ATPase to release energy and make ADP and P, which are the byproducts, start to stockpile until they become to a particular threshold, and then ATPase will start the process over again. Also, you can make ATP from the byproducts through phosphorylation. So you can take ADP and combine it with phosphorus and another amount of energy. Where's that energy come from? The foods that we're eating to make more ATP. And that can occur with or without oxygen. Figure 2.4 shows exactly what we just talked about. There are 3 ways that your body can get ATP. You can you can use whatever your body has stored in its muscle, in the muscle and the liver, but there's not very much of that. Your body also constantly, because there's not much stored ATP, it's forced to make ATP, and that's what eating is. When you eat, you get energy, and that energy is used to create ATP eventually. There's 3 ways that you can create ATP. There's the ATP phosphocreatine system, the glycolytic system, and the oxidative system. We'll talk about each one of those. Now we're on slide 21. The ATP phosphocreatine system. That's where you're actually just burning the ATP that you have for energy, or what you're trying to do is recycle any energy that any of the byproducts of energy that you have, of ATP that you have, to create more ATP. Because you don't store a lot of ATP and because you can't recycle and use it fast enough, the amount of time that you can use that system for is limited to about 15 seconds. So to be quite honest with you, this phosphocreatine system doesn't seem like it's more about the breakdown of energy. It seems like when your body needs to take any energy, any byproducts, and create ATP and recycle ATP, it seems like the phosphocreatine system is more for recycling and making it so that you have an emergency stash of ATP, or you have a stash of ATP in your muscle and your liver, as opposed to breaking that down. The rate-limiting enzyme in this sense is creatine kinase. Phosphocreatine plus creatine kinase is going to result in creatine and phosphorus byproducts plus the release of energy. This is predominantly going to help restore ATP that you burned through walking and through all the energy that you are spending for longer. When you're walking, you're predominantly walking for more than 15 seconds. So when you're walking, you've already burnt out all of that ATP in your muscles, and you got to be using something else. So when you're walking very slowly from here to the bus or whatever, or down from one class to the next, you're burning something other than ATP because you don't have enough ATP to even support that length of activity. So you're using your carbohydrate stores then. All right. So the ATP that you are using during activity is mostly not the ATP phosphocreatine system. It's mostly going to be, and we'll get to that in a second, it's mostly going to be the glycolysis, fast or even the slow glycolysis, but we get to that in a couple of slides. I'm going to skip to slide 26. You can take a look at the animations for slide 23, 24, and 25. The control of this ATP system is controlled by, like we've said before, an enzyme called creatine kinase. It increases when there's an increase in the creatine and the phosphate byproducts. It's a negative feedback system similar to the ATPase system. We're on 27. Slide 27 introduces the glycolytic system. Glycolytic, the glucose, the sugar system, the use of sugar as a system. The glycolytic system doesn't use oxygen. It's anaerobic, so it doesn't need to use oxygen. This whole process that we're going to talk about the glycolytic system is also referred to as fast glycolysis. It happens very quickly, and it happens so fast that the mitochondria doesn't get an opportunity to use any of the sugars to help break down energy. It happens without oxygen, so nothing brings the sugar into the mitochondria. It's an anaerobic system, but the thing here is you can see that before in the phosphocreatine system is 1 mole of ATP to 1 mole of substrate, but here now you have 2 to 3 moles of ATP. It's like 3 times the amount. And not only can you make more ATP because of that, the glycolytic system is going to allow you to do exercise for more than, say, for instance, 2 minutes, from 15 seconds to 2 minutes. Anything after 2 minutes, you're going to have to start to use oxygen, or the slow glycolysis will start to break down. But rather than breaking down ATP, all right, this crazy organism that you are has realized that why break down the ATP stores that I have when I can use a substance outside my body to be able to make ATP for me, which is sugars and carbohydrate, wow, and protein and fat. You can use something outside of your body to create the thing that you need inside your body. So I may as well save what I have inside my body because I can get these stores elsewhere and use the stores elsewhere. All right, not only that, but if I use the other stores, I can create more ATP. It just takes a little bit longer. It's going to allow me to do much better activity. It's going to allow me to do things that last longer than 15 seconds. It's going to allow me to go hunt and go get the things that I need and to go walk and observe and experience the world and stuff like that. So those kinds of activity will, and that energy substrate system will allow me to exercise for 2 minutes. Wow, that's pretty amazing. All right, and it relies on the breakdown of glucose through glycolysis. Again, another breaking down of some process through the lysing, the splitting in half of something. Now I'm on slide 28. The glycolytic system uses glucose, and it converts glucose to ATP. And it starts with this pyruvic acid. I can't go into all of this. I'm not a huge biochemist, so it's going to be hard for me to explain all the minuscule processes. So therefore, I don't expect you to. I'll probably go through these slides a little bit quicker. Anyway, you have a 6-carbon glucose molecule, and those 6 carbons are broken down into 3 3-carbon molecules. Each one of those 3-carbon molecules is called pyruvate. Each of these pyruvates is going to help through a bunch of series of steps with enzymes and enzymes and enzymes, and those are going to break down into eventually ATP. It's going to break down into lactic acid. But that process of breaking down those pyruvate molecules into lactic acid is going to result in the formation of ATP along the series of steps through the process. All right, so eventually that is going to break down into lactic acid, but lactic acid is going to be a limiter to movement and has to get shuttled out of the muscle, or it's going to start to cause the muscles to become acidic, and the acidic nature in the muscle is going to lead to potential muscle breakdown, and that's soreness. So we want to kind of limit the amount of lactic acid that's in the muscles. So the glycolytic system on slide 29, there are pros and cons. The cons is that there's a relatively low ATP yield compared to the slow glycolysis that we're going to talk about next. And not only that, but if I have a lack of oxygen in my system, and I'm not